Okay, so we finished up mitosis, and once again, just as a recap here, mitosis is actually what happens in the vast majority of cells in your body, and what's known as actually your somatic or autosomal cells, everything but your sex cells, right? And in addition, many lower level organisms like prokaryotes, like bacteria, will divide this way asexually without sexual reproduction. That's how very simple organisms actually reproduce, is basically through this basically molecule that'll just duplicate its DNA and then separate and duplicate into two daughter cells. So that is asexual reproduction, if you will. Now we're actually going to be getting into how we produce the cells for sexual reproduction, which is going to actually allow us to have a lot more genetic diversity. We're going to be talking about meiosis. So here's our quick little overview of meiosis. Here, it's similar to mitosis in that the cell duplicates its chromosomes before undergoing this process. But while mitosis preserves the diploid number, that 2n number of cells, meiosis halves it, making it a haploid number. So let's look at this for a second. We have, once again, our starting at the very top, our diploid number here. And what's going to happen is that we once again have chromosome replication. That all looks very similar. But in meiosis 1, instead of lining up along in that metaphase plate, all the chromosomes lining up with their centromeres, this time they're going to line up in pairs, those homologous pairs, that concept we introduced earlier, one from mom, one from dad. And notice they can go on either side. We saw this a bit in genetics already. We already looked a bit at meiosis. So in meiosis 1, once again, there's two nuclear divisions. So here we have meiosis 1, the first. Here's meiosis 2, the second. Two nuclear divisions, making 4, look down here, haploid cells. Haploid N, not 2N. Once again, the whole point here is used to make those sex cells, those gametes, the egg and the sperm. Occurs in sex cells, I should once again be obvious, and once again has 8 phases. Now remember, we had in mitosis, we had PMAT, I said. We had prophase, metaphase, anaphase, telophase. Well, guess what? In meiosis, we're going to have the same 4 things again, slightly different. They're still going to be called the same, because it'll be prophase 1, metaphase 1, anaphase 1, telophase 1 in meiosis 1, right here. And in meiosis 2, down here, we will have prophase 2, metaphase 2, anaphase 2, and telophase 1. So it's all very similar to what we just went over in the last lecture within mitosis. Now, one important part is that in mitosis, we have this chromosome replication in the very beginning, and it happens the one time before it actually separates. Same thing happens here, but notice we don't replicate again down here between meiosis 1 and meiosis 2. There's only this one element of chromosome replication. That's important. So by the time we get through all this, we get through meiosis 1 and we produce 2 intermediate daughter cells, but by the time we get to meiosis 2, we actually have 4 distinct haploid gametes. So here is meiosis 1, and once again, prophase 1, homologous chromosomes, pairs, basically synapse, and crossing over occurs, which there's an exchange of genetic information. I'll get into that more in a minute. But then the metaphase 1 homologous pairs line up at the equator. Anaphase, chromosomes separate and move to opposite poles. So once again, let's go back to just metaphase here. Remember in mitosis, they all lined up like this along that metaphase plate. Well, here they're going to match up. I'm going to go more like this with their homologous pairs at the equator. And in anaphase, once again, in mitosis, anaphase, they started to separate, being pulled this way, right? Those spindle fibers from the centrosome area, where the aster is going out the other way. Central. Remember this we just looked at? Not exactly the best drawing, but I think you kind of remember that. Well, here, instead of once again separating this way, now we're actually separating the homologous pairs, like so. We'll have a better look at this in a second. And then once again, in telophase 1, 2 intermediate daughter cells, each with, in the case of humans, 23 duplicated chromosomes. So they still look like this at the end of telophase 1, as opposed to in a mitosis telophase, they look like this. So let's look at a little bit more specific here. So once again, meiosis 1, prophase 1. Once again, like mitotic prophase, is very similar. However, the homologous chromosomes, once again, when I say homologous, they're coding for the same trait, one from each parent, come together and intertwine, or synapse. So you can see this right here, is that here with how they're all intertwined, these are actually two different chromosomes intertwined here, homologous chromosomes, one from mom, one from dad. And this part, this place right here, because of the way they're coiled up, things can start crossing over. So even though we have one distinct chromosome from mom and one from dad, at this point they intertwine and big pieces of DNA can kind of flip over to the other side. And this once again allows for even more genetic diversity. So at this stage, once again, each chromosome consists of 2 sister chromatids, and each synaptic pair of homologous chromosomes contained 4 chromatids, a tetrad. Sometimes chromatids, once again, break at corresponding points. That's what I'm talking about with this crossing over. And they exchange, and here's the important part, equivalent pieces of DNA. Now, the chromosomes remain joined at points called chiasmata, where the crossing over occurs. So once again, I'll show you an animation a little bit, but you'll be able to see this concept of crossing over. So, still moving in meiosis 1, moving from that prophase 1 to metaphase 1. And now, once again, we're starting to see more differences between mitosis and meiosis. Here, rather than lining up all along the center, all the chromosomes, here the homologous pairs independently align. So once again, we could say, let's say for just this argument's sake, blue is from dad and red is from mom. We can look up here, and here is one chromosome, mostly from dad, but wait a minute, what's this red piece? Well, see, this is where crossing over occurred, here and here. This is representing where crossing over occurred over here in prophase 1. Same thing down here. We're saying this chromosome mostly came from mom, but because they intertwined over here, some crossing over occurred at this point, and these 2 equivalent pieces crossed over. So that happened in prophase 1. and metaphase 1, we line up, but we line up the homologous chromosomes. And then we enter anaphase, where the homologous pairs separate and are pulled to the opposite sides of the cells, called disjunction. And each daughter cell will have a unique pool of alleles. Remember the alleles, remember when we talked about genetics, the genetic coding for alternative forms of the same trait, right? So each daughter chromosome will have a unique pool of alleles from a random mixture of maternal and paternal origin from mom and dad. Then telophase, coming near the end of meiosis 1, The nuclear membrane forms around each new nucleus, and the cell divides into two daughter cells, each of which receives, once again, what? The haploid number. We started in this over here. Over here we started diploid number 2n. In this case, in this example, there's 4 chromosomes here. But notice how many we have over here. What's going to be happening at the end of telophase, we're going to have 2 chromosomes in each of these. They are the haploid number, right? So this, once again, is not the end. This is the only end of meiosis 1. We still have like a cytokinesis, and there's a slight pause here. So these are actually really known as intermediate daughter cells. And once again, we have interkinesis, they call it here. We have the separation. We do have haploid, but they're still duplicated haploid. They're still like this. They still have double their DNA. So they still consist of 2 chromatids. So once again, let's watch a little animation from meiosis 1.
Meiosis follows interphase after the chromosomes of a diploid cell have replicated. DNA replication occurs during S phase. After the 2 successive cell divisions of meiosis, a single cell will produce 4 cells, each having the haploid number of chromosomes. Prophase 1 of meiosis 1 begins with the condensation of chromosomes and the vesiculation of the nuclear membrane. Centrosomes, which duplicate at the beginning of meiosis, begin moving apart, and the spindle fibers begin forming. Early in prophase, synaptonemal complexes form, and the homologous chromosomes are seen to align next to each other to form a structure called a bivalent. A bivalent is composed of 4 sister chromatids. Crossing will occasionally occur between homologous chromatids within a bivalent. During prometaphase 1, the spindle apparatus continues to form, and bivalents become attached to kinetochore microtubules. One pair of sister chromatids is connected via kinetochore microtubules to one pole, while the homologous pair of sister chromatids is connected to the opposite pole. The complete assembly of the spindle apparatus occurs during prometaphase 1. The chromosomes align at the metaphase plate during metaphase 1. It is important to notice that bivalents are aligned along the metaphase plate rather than individual pairs of sister chromatids, as in mitosis. Anaphase 1 involves the separation or disjunction of homologous chromosomes, with each pair of sister chromatids moving to the opposite pole of the cell as the kinetochore microtubules shorten. When the conjoined sister chromatids reach the poles of the cell, they detach from the spindle apparatus, and the nuclear membrane begins to reform. A cleavage furrow forms, and cytokinesis ultimately results in 2 cells, each having the haploid chromosome number, with each chromosome consisting of a pair of sister chromatids. This completes meiosis 1, and after a period of interkinesis, the 2 daughter cells start the second cell division cycle called meiosis II.
Okay, so what is crossing over? So with crossing over, not only are we getting the genetic variation of being able to separate, in our case of human beings, the 46 chromosomes, 23 from mom, 23 from dad, and just randomly being able to separate which one's from mom and which one's from dad. So imagine, well, it's not quite the same as a deck of cards, but pretty close. Imagine all the, there's 52 in a deck of cards, all the red cards, diamonds and hearts are from mom, and the black cards, the spades and the clubs, are from dad, and then just randomly separating who's going to get what of black cards versus red cards. But there's also this concept of crossing over, where we actually get even more exchange of genetic information between non-homologous sister chromatids during a synapse. Once again, this occurs during prophase 1 of meiosis, and it once again increases that genetic variation, and we saw this already. So we have the the homologous chromosomes here, right? Each with their own sister chromatids, and they basically cross over and exchange DNA information. So once again, it allows for even more genetic variation. And I really like this animation, straight from your book once again, so let's watch this one.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase 1 of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, one parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now consider the case where there is crossing over in the region between the 2 genes. Since the crossing over is only between 2 individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other 2 will be recombinant, with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
So once again, it's all about being able to create more genetic diversity. Now we get to mitosis 2. And the good thing here is that it's very much like mitosis. So if you understood mitosis, it's going to be very, very similar. And that the big exception here is that it's not preceded by chromosome replication. So once again, we have prophase, where the chromosomes will condense again. Then we have metaphase, once again prophase 2, metaphase 2, where they align at the equator. Here, the anaphase 2, the sister chromatids separate to the opposite poles. And then finally, once again, we end up with 4 haploid daughter cells. In the case of humans, 4 daughter cells with 23 unduplicated chromosomes in each. So here's prophase 2. The cells have 1 homologous pair. Cells have 1 chromosome from each homologous pair. That's what I meant to say, excuse me. And now, remember we started with 2. Then we enter metaphase 2, so they line up at the equator. Then we have anaphase, they separate. Once again, this should look very similar to mitosis at this point. Telophase 2, the spindle disappears, once again just like mitosis, and cytokinesis will take place. And what do we end up with? We started with 2 haploid cells over here on the left. Once again, 2 haploid cells, right? And now over here we have 4 haploid cells. So once again, this looks very much like mitosis. The big difference is we're starting with haploid cells, and we don't have any need for any DNA synthesis reproduction beforehand. There's no chromosomal replication. And the key is, once again, we end up with 4 haploid daughter cells. And when we're haploid, we know we're talking about those sex cells, right? This is the type of replication to create the sex cells, the sperm and the eggs, spermatogenesis and oogenesis. So now, once again, a little animation on prophase 2.
At prophase 2, the chromosomes condense again. and the nuclear membrane vesiculates. During prometaphase II, the spindle apparatus forms, and the sister chromatids become attached to kinetochore microtubules. In this case, a pair of sister chromatids is attached to both poles, not just one pole as in prometaphase I. At metaphase II, the sister chromatids are aligned along the metaphase plate, with each chromatid being attached to one pole. The sister chromatids separate or disjoin and begin moving to the poles of the cells during anaphase II. During telophase II, the nuclear membrane reforms and chromosomes decondense. In animal cells, a cleavage furrow is formed that causes cytokinesis or cellular division. As cytokinesis is completed, meiosis II results in the formation of 4 haploid cells. Thus, meiosis begins with a diploid mother cell that has replicated its chromosomes and ends with 4 haploid cells containing one set of chromosomes each.
And once again, it's all about genetic diversity here. And so let's look about this random orientation of chromosomes during meiosis.
During metaphase I of meiosis, each joined pair of homologs lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the maternal or paternal homolog may orient toward a given pole. The number of possible chromosome orientations equals 2 raised to the power of the number of chromosome pairs. In this hypothetical cell with 3 chromosome pairs, 8 2⁵ possible orientations exist. Each orientation produces gametes with different combinations of parental chromosomes.
Now, in that example, once again, it was 2⁵. As far as that haploid number. Remember, what are we again? Oh yeah, our haploid number is 23. Think how big that number is.